Without a specific set of molecular helpers, the chemistry inside you would move so slowly it would take decades to process a single bite. To survive this, you need a physical way to grab those exact molecules and force them to react right now. This is where enzymes come in. They are biological accelerators, working around the clock to keep the millions of chemical reactions inside you moving at a rapid, life-sustaining pace. Each enzyme has a specially shaped pocket designed to fit only one specific type of molecule. When they fit together, like two puzzle pieces snapping into place, the reaction happens instantly, and the finished product is released. This exact fit is why your body functions so smoothly. By choosing exactly which reactions to speed up, these molecular machines act as biological micromanagers. But these workers are surprisingly delicate. To hold their shape, they require a perfectly stable environment, meaning the temperature and acidity levels around them have to be just right. If the heat rises too high or the acid levels shift, the enzyme structure literally unravels. The docking pocket collapses and it can no longer grab its targets. This process is called denaturation. It's the moment the machine breaks for good. Your body expends massive energy shivering, sweating, and balancing its internal chemistry for one main reason: to keep these fragile machines perfectly shaped and functional. To understand how well a specific enzyme is performing, scientists measure its speed by looking at how much raw material is available for it to work on. This chart tracks that speed. At first, as we provide more material, the enzymes process it easily. They work faster and faster, and the reaction speed shoots up. Eventually, every single enzyme is busy. Even if you pour in more material, there are no open spots left. The speed stops increasing, and our curve flattens out. We draw a line right across that flat top to mark the maximum speed limit for that group of enzymes. In science, we call this Vmax. Now look at the exact halfway point on that speed chart. By measuring how much material it takes just to get to that halfway speed, we find the enzyme's grip strength, a value called Km. A lower number here means the enzyme is incredibly efficient at snatching up its targets, even in a crowd. This chart compares two different versions of these sugar-snatching workers. You can see they have completely different speed limits and grip strengths. One hits its limit early, while the other takes much more material to reach its higher potential. Mapping these two numbers tells us exactly how efficiently an enzyme works. It gives us the mathematical foundation needed to see how the body maintains its balance at a microscopic level. Sometimes a reaction needs to be slowed down or stopped. To do that, we use inhibitors, chemicals designed to act like a wrench thrown into the machinery. A competitive inhibitor is a shapeshifter. It looks enough like the real target to sit in the docking pocket, but it won't react. It just blocks the way. To clear the path, you have to flood the system with real material until the intruders are simply crowded out. A non-competitive inhibitor is more subtle. It attaches to a different spot and warps the enzyme's frame from the outside. Because the pocket is now the wrong shape, adding more material doesn't help. The top speed limit of the whole system just drops. Most of the medicines we use today are actually custom-built inhibitors. They are designed to safely block specific enzymes to lower cholesterol, stop pain, or kill bacteria. Your body uses slightly different models of these workers in different places. They do the same job, but they carry unique structural tags depending on whether they belong to your heart, your liver, or your muscles. This physical tagging system is vital when someone is critically injured, such as during a heart attack. When heart muscle cells are damaged, they break open. All those uniquely tagged heart enzymes spill into the bloodstream. Doctors don't have to guess what's happening. By taking a blood sample, they can look for those specific heart enzymes. They trace the leak back to the source to confirm exactly which organ is struggling. Studying enzyme structures and how they break moves us beyond abstract chemistry. It is the language doctors use to pinpoint exactly what is happening deep inside the body. From the precise fit of a target molecule to the speed limits we plot on a chart, and the chemical wedges we use to slow them down. By tracking how these tiny machines work, fail, and leak, we make the invisible mechanisms of life visible to the people who save them.